All right, team. Here we are again. The dogs are going crazy. Jeez, I don't know what's going on outside, but here I am sitting in my little room about to teach you your last lesson for the first test for Bio 111. Okay. So this is going to be just mostly chalk and talk, honestly. Just going to open up the Sketchpad and we're going to talk a little bit about what we want to learn today. Movement of substances in and out of cells. So, there's a lot to this. There are some laws that govern the way stuff moves in and out of cells. And we're going to talk about those. There are laws about how molecules move specifically. And then we'll talk about how larger things get in and out of cells. Okay. So primary law, one of the primary laws of how things move is the law of diffusion. And the law of diffusion says molecules move from higher to lower concentration. Period. It doesn't specify what the molecules are. You could say stuff moves because it's all matter. All matter follows this law. It is a law. In science, if something is a law, that means you can reproduce the results over and over and over without exception. So that's something else to think about. So what does this mean? Well, what it means is if you have a substance where there's a higher and a lower concentration, then the molecules are going to move from that higher concentration to that lower concentration. Try to imagine, if you will, if I had a beaker here that was full of water. Or not quite full of water. And then I took some red food coloring and I drop a drop of red food coloring into the, weird, I wonder what happened there. I got some kind of marker or something. I don't know what happened. That's odd. Oh, I see what happened, sorry. Let's say I dripped a drop, one single drop of red food coloring down into this beaker of water. Okay, now I have a concentration gradient, right? I have this dye, this drop of dye that fell to the bottom of the water. Over time, the dye molecules are going to start to diffuse. They're going to start to move through the water from a higher concentration to a lower concentration until at some point there is no longer a concentration gradient and this dye molecules are equally distributed throughout the vessel. I don't know how long that's going to take, but it will over time happen. Now, in this case, you have a liquid diffusing through a liquid, but you could have a liquid diffusing through the air. Have you ever experienced someone that wears a little bit too much cologne, right? So they put this liquid cologne on their body and everywhere they go, that cologne is diffusing into the air, right? Okay, so at some point, the dye molecules now are going to be equally distributed throughout the water molecules. At that point, we say equilibrium has occurred. And when equilibrium occurs, that just means there's no longer a concentration gradient. So equilibrium takes time. Now, different states can diffuse, gases can diffuse through gases. We said, liquids through liquids, liquids through gases, etc. Okay, so that's diffusion. The next law that we want to talk about is the law of osmosis. And the law of osmosis is a kind of diffusion. Osmosis is a kind of diffusion. The law of osmosis says it specifies what's doing the diffusion, the diffusing. It is specifically the diffusion of water. And we're going to add to that, we're going to say through a semi-permeable membrane. What in the world are we talking about? A semi-permeable membrane. Well, you know what permeable means. It means stuff can pass through. If it's semi-permeable, that means some stuff can pass and some stuff can't. So for example, a coffee filter is semi-permeable, right? You put your coffee in it and the grounds stay in the filter. It's not permeable to the grounds, but the water can pass through to make your coffee, right? What's the membrane? What's the semi-permeable membrane of living cells? Well, that should be clear. What we're really talking about is the plasma membrane, right? So when we talk about osmosis, we're talking about water moving in and out of cells through that plasma membrane based upon the law of diffusion. Let's look at how this might work. Suppose we have the following scenario. Suppose I said, okay, we have 3 cells and we'll let them be animal cells. So there's no cell wall, there's just a cell membrane. And let's say that the inside of one of the cells is 1% salt solution. And let's say the inside of another cell is 3% salt. And then another is 5% salt. So we have 3 different cells. They have different concentrations of salt inside of them. And let's just say we bathe all of these cells in 3% salt. And this is all salt. Okay, you get the idea. So now the question is, based upon the law of osmosis, water is going to move in one direction or another based upon a concentration gradient. Okay, so if we look at this first cell, we have 1% salt on the inside and 3% salt on the outside. It's important to understand what a solution is. A solution is equal to the solute plus the solvent. And there are many different kinds of solvents. Solvents are things that dissolve solutes. And what's the solvent of life? What is life dissolved in? Water, right? In this case, the solute is salt. So the solute is what's dissolved in the solvent, and the solvent is what dissolves the solute. Together, they make up a solution, okay? So here's the tricky thing. You know the water is going to move from higher concentration to a lower concentration. What you have to understand is that the salt doesn't move. The salt on the outside, this 3% salt on the outside is stuck there. It can't move through the membrane. Only water can move through in our example. And the 1% salt on the inside is stuck. So don't get sucked into thinking 3%, 1%, it's going in, because that's the salt concentration. What you need to do is look at the salt concentration and figure out the water concentration. If it's 3% salt on the outside, well, that means it's 97% water, right? That's the solvent. And on the inside of this first cell, we'll let this be cell 1 and cell 2 and cell 3. On the inside of this first cell, you have 1% salt, so you have 99% water. Okay, which way is the water going? Based upon osmosis, it's going from a higher to a lower concentration. So in this cell, water is going to leave the cell like this. Okay, following that concentration gradient. What about this one? We have 3% outside, 3% inside, so we got 97 and 97. This is equilibrium, right? This cell is at equilibrium. It's not, the salt's not going in or out. It's going to stay right where it is. And then what about this last one? Well, we got 5% salt on the inside, so we got 95% water. and we got 97% water on the outside, higher to lower, water's going to go in the cell until it reaches equilibrium. So by knowing the concentration of water on the inside and the outside, we can predict what direction the water is going. This first scenario is a good example of why you can't put a freshwater fish in salt water, right? If you put a freshwater fish in the salt water, all the water leaves its cells, right, going toward that. Salt sucks. If you just remember salt sucks the water. So the higher the salt concentration, that's the direction the water is going. See here, the water's going in. This is why you can't put a freshwater fish in salt water. It will die. This is why you can't put a saltwater fish in fresh water. The water will flood its cells, and that's equally as damaging to the cells. So this is good because we're at equilibrium. Water's not going in or out. Okay. Okay, so with all that said, what about a salmon? What about a salmon? A salmon is born in fresh water. It swims out into the ocean, into salt water, where it lives its life until it's ready to reproduce. And when it's ready to reproduce, it swims back, and believe it or not, it always finds the same creek that it was born in. It can smell the creek. How crazy is that? It always goes back to the same creek, and then it lays its eggs. Well, how can a salmon go from saltwater to freshwater and back? What must it be able to do? It must be able to regulate the salt content in its cells. And what it does is it literally can pump salt in or out of its cells to maintain equilibrium as it goes from one concentration to the next. Otherwise, it wouldn't survive. Okay. Beautiful. So this is how many molecules, including, well, this is how water moves in and out of cells readily. Okay. Both the first and the third cell can be damaged by this happening. This is particularly damaging. It's called plasmolysis. You don't have to know that word. And this is called crenation. So there's, there are consequences to this this salinity or this concentration thing. Okay, so that explains a lot, really, about how stuff moves. Think about simple diffusion. This is how oxygen gets into your blood and carbon dioxide gets out of your blood. It's just a concentration gradient. And the CO2 goes into the blood because there's a concentration gradient there, a higher concentration in your lungs and a lower concentration in the blood. And so it goes in that direction. The cool thing about diffusion and osmosis is that they require no energy. How weird is that? What I'm saying is you can move stuff without putting energy in these examples, as long as you're going with the concentration gradient. Okay. So now we're going to redraw our, what we want to think about now is how do big things get in and out of the cell, or bigger molecules, we might say. Well, let's draw our phospholipid bilayer, because really in order to get in and out of the cell, they have to get through this membrane. And then we're going to draw a protein that's embedded and goes all the way through the phospholipid bilayer, and then we'll draw a little bit more membrane. And you remember these phosphate heads are in the water and the fatty acid tails are sticking in either direction like that, okay? So this protein is called a transport or carrier protein. It has a very specific function, and that very specific function is to move a particular molecule in or out of the cell. Okay, so let's say we have this scenario here. Let's let this represent a glucose molecule, just for fun. Okay, and so what then can happen is if you have all this glucose out here, right, remember proteins have a specific shape, and here's this little active, I don't want to call it an active site because it's not an enzyme, but this molecule fits in there, this glucose molecule, and it gets shuttled into the cell like this. It can go in either direction. Most of these transport proteins can transport in or out. Now you'll notice that a transport protein is specific to a specific molecule that it's going to move. It's not just random. It can't move a bunch of different molecules. Okay, so let's think about some scenarios of this and whether or not they would take energy. I guess that might be something that you would want to think about. Okay, let me get rid of this larger picture and let's just draw it like this. Let's just draw the carrier protein like this. And let's say this is out and this is in. And so let's say that there's all this glucose here on the outside of the cell. And there's only a little bit on the inside. The law of diffusion says that the glucose wants to go in which direction? Well, there's more outside than inside, so there's a concentration gradient that's forcing it in this direction. And so in this scenario, the glucose, its movement is being facilitated by the protein, but no energy is required. In other words, diffusion will just allow this to happen without any energy because it's going with the concentration gradient. We call this passive transport. Or facilitated diffusion. Facilitated because there's a molecule, a protein involved to facilitate the passage, but other than that, it's going with the concentration gradient, and so it doesn't require any energy. It just happens as long as this protein is there to help. Okay, so that's one scenario. What if we have this different scenario? What if it's like this? What if there's a little bit of sugar on the outside and a lot on the inside? Okay. The law of diffusion says the sugar wants to go out, right? But let's say this cell needs more sugar for whatever reason. It still is trying to get more sugar. Well, is it possible to go against the concentration gradient? Is it possible to move the sugar in the wrong direction, essentially? And what I might compare it to is you decide to go paddleboarding on the French Broad River and you put in, at sandy bottoms and you're just floating for a half an hour down the river and you're just floating along. That's like passive transport, right? You're going with the flow, with the concentration gradient. No energy is required, okay? But then what happens if you need to get back and you turn that paddleboard around and you start going against the current? What best you be doing? Well, you best be paddling, right? You must put energy in to go against the flow or against the concentration gradient. We call this scenario active transport. And in active transport, you're going against the concentration gradient. And I guess up here we should say with concentration gradient. And this requires energy. Just remember, when you're active, you're using energy, and when you're passive, you're not. Okay, so what are the implications of this? Well, we need to put in energy, and the energy is in the form of this stuff called ATP. We see this molecule ATP being converted to ADP. And I don't expect you to know exactly what's going on here, but the reality of it is we're going to learn a lot more about this, but this is the energy that is used to move, to do this process of moving these molecules against the concentration gradient and into the cell. Okay, so active transport uses energy, passive transport does not. Okay, that's all good. And now the only last thing to think about before we're literally done with the unit is how do large things get in and out of the cell? That might be something to think about. Let's get rid of this. By large things, I mean things that aren't molecules, things that are larger than molecules. And there's a process for doing this, of course. Here's an example. You have an immune system. In your immune system, you have these cells, they're called killer T cells. And we'll just use this as an example. Killer T cells are white blood cells, lymphocytes, and their job is to roam around the body and look for things that don't belong. And when they find something that doesn't belong, they literally engulf it or eat it. And this process of cellular eating is called endocytosis. Literally, it means cell eating. Okay, so let's draw a little bacteria. There's this little guy, he's a little bacteria with a smiley face. He's happy, okay? And here's a killer T cell. Killer T cell comes along. It is able to recognize that this bacteria doesn't belong based upon its proteins on its surface. And we're going to talk a lot more about that when we talk about the immune system. Okay, and viruses and whatnot. But suffice it to say, what will happen is when it recognizes this as being foreign, it will do this. It will form this little evagination of its membrane like this. Okay, and then we look at the next picture. Might look something like this. And now the little bacteria is frowning because it's so scared. And then ultimately you end up with this. Now you'll notice that because of it's in this sort of evagination of the membrane, this bacteria it's not freely in the cell. It's in a vesicle, right? It's in a little bubble of membrane. It's trapped. And then guess what happens next? See how we build on all the stuff we've learned. Along comes a lysosome with its digestive enzymes. Remember, a lysosome is a vesicle with digestive enzymes, and it will literally just dump them in there. And then that little bacteria will be digested. This is endocytosis, or cellular eating. Another word you might come across, which means the same thing, is phagocytosis. So either of those words are correct for cell eating. What about exocytosis? Well, just the opposite would be exocytosis. And so you might have a cell, I guess if endocytosis is cell eating, then exocytosis is cell vomiting. Okay, you might have a cell that's got a bunch of waste products in a vesicle, and it wants to get rid of that. Or even better example, secretory substances. Think about the mammary glands, right? They produce milk in cells, and they have to get that milk to the outside. So this vesicle full of milk will come and merge with the edge of the membrane like this. And then ultimately it gets released to the outside. Little baby gets its milk. Okay. Draw a nucleus in all these cells just for fun. So endocytosis, cell eating, also known as phagocytosis. Exocytosis is cell regurgitation. It's a way to get rid of, to get products to the outside or to get rid of waste. Okay. There you have it. That is the end, I believe, of the unit. So just to be clear, we are now finished with the information for the first test. Okay. And what I want to tell you is what I want to tell you is, you know, there are these videos, and then you have your textbook, and hopefully you've been reading the material in your textbook. You have, you'll have you know, the test will open very soon. Once the test is open, you'll have, I think, a week to take the test. So I'll send you an email with those dates. Okay. But, you know, this completes the information for the first unit, for the first test. I hope you've enjoyed this lecture, and I will see you next time. Bye-bye.